Now, I'm pleased to share how you can use the Restore and Renew web tool to improve the short and long-term success of your biodiversity and carbon plantings. Now, I'm a practitioner engagement facilitator at the Research Centre for Ecosystem Resilience, or RESA for short, at the Botanic Gardens of Sydney, and a key part of my role is to help ensure that practitioners such as yourself undertaking restoration are aware of the relevance of our research and understand how to apply it on the ground. Now, much of what I'm presenting today is based on research by my colleagues Maurizio Rossetto and Marlene Vandermeer and others at RESA, so I'd like to acknowledge them up front. Now, I'd also like to acknowledge the traditional custodians of the land where our research centre is located, the Gadigal people of the Eora Nation, and I also honour all the traditional custodians of the lands where we've collected material from for this research, and I'm grateful for the care and wisdom they demonstrate in caring for country. Now, I've included QR codes throughout the presentation, linking to further information. This one goes to the RESA homepage. So, just briefly, we're a team of 20 researchers based at the Royal Botanic Gardens in Sydney. And here's some of the team planting a Wallamai pine, which is just one of the many species we're gaining insight into using genomic tools. So we all have a common interest in guiding restoration and conservation of resilient ecosystems, hence the name, through large-scale genomic projects. Now, to give you an idea of the scale, our team and collaborators have collected leaf samples from more than 89,000 plants across Eastern Australia for more than 2,000 species, and already 54,000 of these plants have had their DNA sequenced. These all contribute to what we call our genomic knowledge infrastructure, which is effectively one whopping big data set which can be applied to answer all sorts of questions. So just briefly, here's some of the research or areas where our research is applied. I'll be talking today about how about the Restore and Renew web tool and how it guides seed sourcing for restoration. But we also, for example, provide advice to the New South Wales Saving Our Species program and to large-scale collaborative projects such as the Big Scrub Rainforest Conservancy Science Saving Rainforest project. And these QR codes all link to more information on these key research areas. So this presentation is all about empowering you to know what to ask for when sourcing plants for your biodiversity and carbon plantings. Now, I'll highlight why genetics is relevant to revegetation and demonstrate how you can use the Restore and Renew web tool to plan where to source your plants from in order to safely maximise genetic diversity. Now, in the short term, you're going to be constrained by what the local nurseries are growing. But in the long term, thanks to the very worthwhile North Coast Seed Bank project, you'll be able to source seeds specifically for your project and commission nurseries to grow your plants from that seed. So this presentation will empower you to design your own seed sourcing strategy. So restoration is a long-term gain. None of us want to be wasting our time. We want our restoration efforts to be successful in the long term. Now, a lot of the focus these days is on quantity, the number of plants, the area of plants. But for all our efforts to be well spent, we need to also focus on quality. Now, the quality of your restoration plantings matter. Now, to be successful, revegetation needs to be self-sustaining. Now, in order for restoration to be self-sustaining, or for restored populations to be self-sustaining, it needs to be made up of functioning populations of its component species. So, and all these populations must reproduce and also need to be able to adapt to change. Now, for populations to be able to reproduce and adapt to change, they need genetic diversity. Populations can't reproduce well or adapt to a change if they don't have genetic diversity. So let's jump into a bit of a genetic refresher so that the following conversations regarding inbreeding and outbreeding depression and other concepts is a little bit easier to follow. So DNA is the molecule that carries the genetic code of an organism and is organized into genes. Now, these genes are sections of DNA that give instructions for making proteins and determine how an organism looks or functions. For example, whether or not it is short or tall, able to cope with increased temperature, or whether it is resistant to a disease like myrtle rust and so on. Now, alleles are alternative forms of a gene, and at a given location in the genetic code, each individual has two alleles, one from each parent. Now, these alleles can be the same, homozygous, or different, heterozygous. So the genetic code or DNA sequence is different between individuals within the same species. This is genetic diversity. Now, the more genetic diversity in your planted population, just by chance alone, the greater the probability of your restored populations possessing a trait that enables a population to adapt to change. For example, the greater the genetic diversity, the greater the chance of at least some individuals having the genes that enable a plant to tolerate increased temperature or be resistant to myrtle or a disease like Phytophthora or myrtle rust or whatever the next big devastating plant disease is. So whereas just by chance alone, if your planted population has low genetic diversity, it has less chance of having the genes to enable adaptation to change or some future threat. So it has low adaptive potential and increased extinction risk. Now, another consequence of low genetic diversity is reduced fitness or inbreeding depression. Now, a population with low genetic diversity can become so inbred that seed are no longer produced or seedlings may be weak and unlikely to thrive. Now, I'll take a moment to explain, I guess, the background, the genetics behind this. So this is due to the expression of deleterious recessive alleles. Now, a deleterious recessive allele negatively affects an individual's fitness when homozygous. In other words, when an individual has two identical copies of allele, but they're harmless when masked in the heterozygous state. In other words, when an individual has two different versions of an allele, the plant exhibits the traits of the dominant allele. Now, the key point to note here is that as genetic diversity decreases, the probability of a plant being homozygous or the same for a particular allele increases because both parents are more likely to have a similar DNA sequence. And therefore, the chance of a deleterious recessive allele being expressed increases as genetic diversity decreases. Whereas when genetic diversity is high or heterozygosity is high, because each parent is more likely to have differences in their DNA sequence, the deleterious recessive alleles are masked by the dominant allele. So this is particularly relevant for large-scale restorations where vast numbers of eucalypts may be planted from seeds sourced from the same tree. Now, this is not uncommon. Now, these plants are close siblings and their offspring are more likely to express deleterious recessive alleles. Now, eucalypts are obligate outcrossers. They don't do well when mating with close relatives. So much so that the inbred seed is often not viable, although it may be produced, it isn't viable or it doesn't germinate or seedlings don't do well. But eucalypts love to hybridize. So if you have siblings planted at your restoration site, the seed that they produce is more likely to be hybrid than pure and not what you're intending to plant. And there's a QR code there for an article on how genomics can improve eucalypt restoration. So in summary, the more genetic diversity a population possesses, the more self-sustainable it will be in the short and the long term. So here we have a hypothetical North Coast carbon planting. Now we all know diversity of species is important. I understand you're required to have a minimum of 8 tree species and 6 small tree or shrub species, so at least 14 species in total. But for your population to be self-sustaining, you need to not only have diversity of species, but diversity within species, so that your population is functioning self-sustainable in the long term. Now, to maximise the health of your individual plants, you also need to have genetic diversity within individuals. So you don't want the individuals that you're planting to be highly inbred. So the diversity within your species facilitates resilience or ability to adapt to change, and the diversity within individuals avoids inbreeding depression. So now let's talk about how genetic diversity can arise and the consequences of it in your restoration planting. So low genetic diversity can arise from both habitat loss and narrow seed collection. So here we have a genetically diverse natural population of a single species, habitat clearing happens, and just by chance alone, we know that when we lose individuals, we lose genetic diversity. So someone comes along, collects seed from just a few plants, which is not uncommon, and their narrow collection amplifies this loss of genetic diversity. Now, in the short term, what this means is that we may get inbreeding and no recruitment. Now, and it also means that once those individuals die due to old age, there's no new recruits to ensure the population survives in the long term. Now, say we come along and we slightly increase the number of individuals or the number of individuals that we collect seed from. So in the short term, you may actually think that your restoration planting has been successful because you can see natural recruitment, but then say a new stress comes along, such as climate change or a new disease, and just by chance alone, the population may not have the genetic makeup that enables the population to adapt to that change. Low genetic diversity, low adaptive potential. Now, for your plantings to be a success in both the short term, you want to source seed so that you capture maximum genetic diversity. So your genetically diverse, so you have your genetically diverse seed collection, which results in a genetically diverse restored population, which exhibits normal recruitment, and over long term, you've actually increased the chances of your population being able to adapt to change because you've got a increased genetic diversity and a greater adaptive potential. So in summary, you want to avoid your plants being too closely related because it can result in inbreeding depression, hybridization in preferentially outcrossing species like eucalypts, and can result in your plantings being unable to adapt to change. Now, another consequence or negative consequence I've not yet spoken about, which is particularly relevant to those of you with remnant vegetation on or not near your property, is the loss of genetic diversity in natural populations due to genetic swamping. So imagine you plant thousands of closely related plants and nearby is a remnant with a small number of individuals of the same species. Now these natural individuals may be genetically distinct from those being planted and may have rare alleles that are adapted to the location and something you want to conserve over the long term. But just given the large number of planted individuals over generations, the rare alleles will likely be lost because of the greater number of planted individuals contributing to the gene pool. Now, we not only want to avoid our plants being too closely related or having too little genetic diversity, I guess we want to maximize genetic diversity, but we don't want to go too far. So we want to also avoid our plants being too unrelated, because it can result in a loss of local adaptation and outbreeding depression. So outbreeding depression is caused by the breakup of co-adapted gene complexes. So imagine you have a group of genes that, when they occur together, confer some sort of fitness advantage or adaptive capacity. So these genes evolve together, they co-adapted, and together they form a co-adapted gene complex that has some sort of evolutionary advantage. So it's always present because all individuals have it in that genetic neighborhood or in that genetic lineage. But if you've got two individuals that would not usually cross naturally, the DNA can be so different that these co-adapted gene complexes may be broken. So when maximizing diversity, we don't want to go too far. There's a limit. Now, the Restore and Renew web tool can help you avoid planting individuals that are too closely related, and it can also help you understand how far is too far when you're trying to maximize genetic diversity. Now, the Restore and Renew web tool helps guide you to safely maximize genetic diversity to avoid inbreeding, but it helps you know how far to go so that you don't avoid outbreeding depression. So it guides seed sourcing for resilient restoration, and it identifies seed collection areas based on genetic information. So the current version of the web tool, there on the right, QR code, is based at restoreandrenew.org.au. But later on in this year, we'll be launching version 2 of the web tool in collaboration with ADAPT New South Wales. And it'll look different and have a new home. Now, before we talk more about genetic neighborhoods, I want to touch on the idea of local provenance. What is it and why have we been told to use it? So for decades, best practice has been to plant local provenance. Now, it's a generalization aimed at capturing material adapted to local conditions and also avoiding outbreeding depression. But we all know that local conditions are not stable, and we also know that it's actually not easy we can't generalize about how far is too far. So when I ask this question, what is local provenance at workshops, I get all sorts of answers. So 4 kilometres, 40 kilometres, a catchment, a local government area. But what we know is we can't actually generalise. Now, as an aside, the evidence now suggests that inbreeding depression is of more concern than outbreeding depression. And we no longer need to generalize. A genetic neighborhood, it has the benefit of being based on genetic data rather than generalizations. So I'll briefly take a moment to define a genetic neighborhood. So it's an area within the distribution of a species where there are no restrictions to gene flow other than the distance between individuals, such that alleles or a variation of a gene or a different version of a DNA sequence can be shared between individuals within this area given sufficient time. In the web tool, we also call this a genetic collection area. So what this definition means is that you're not going to break apart those co-adapted gene complexes within a genetic neighborhood because the genes are already being shared. These co-adapted gene complexes are not going to be broken up. So inbreeding individuals within a genetic neighborhood can be crossed without risking outbreeding depression. So the Restore and Renew web tool clearly defines geographic areas or genetic collection or genetic neighborhoods or genetic collection areas where seed can be safely collected without risking outbreeding depression. So let's just highlight how, we can't make generalizations we can't predict a safe distance to be able to mix material. So here's a study on 5 similar looking acacias, or morphologically similar, all occur in similar environments, but they all have very different genetic neighbourhoods. So we have a restoration, here's a publication here, you can click on the link to see the paper, but we have a restoration site west of Sydney, and looking at the genetic neighbourhood or genetic collection area, it varies considerably. So it's the grey area. So for Acacia linifolia, longifolia and terminalis, seed can be safely sourced from a relatively large geographic area to maximise genetic diversity. Whereas for Suaviolans and Ulicifolia, which I'm sorry you can't see, but it's actually a really small circle, the area over which seed can be safely sourced without risking outbreeding depression is much smaller. So we can't generalise, but nor do we need to, because genomic tools have become more affordable and accessible, and we can actually more easily get real solid genetic data which tells us how far is too far. So let's look at genetic collection areas downloaded from the Restore and Renew web tool for a hypothetical site near Kempsey. So the scale is consistent. That's the east coast of New South Wales. So the genetic collection areas vary considerably. For example, Angophora castata, you can safely source seed from the Queensland border south to Jervis Bay, whereas with Calicoma ceratifolia, the genetic collection area is far smaller. And here we have genetic collection areas for 3 other species, all very different. So here's a list of species on the web tool so far. There's more than 80 species, and when version 2 of the web tool is launched later this year, we'll have close to 100 species. And over the coming years, we hope this will grow to many hundreds. Now, the web tool can also help you incorporate climate-ready material into your plantings. So here we have a hypothetical restoration site between Kempsey and Coffs Harbour. Now, the web tool identifies current climate matched areas. These are in blue. These are areas where the climatic conditions match that of your restoration site currently. It also identifies future climate matched sites. So in these areas, in the future climate match sites, they're currently experiencing climatic conditions similar to what your restoration site is predicted to experience in the future. So if you source seed from these areas, you know your plants are climate ready because they're from plants that are already experiencing the future climatic conditions. So in summary, to maximise genetic diversity, or to safely maximise genetic diversity and climate readiness of a restored population, you use seed collected from multiple individuals across multiple sites across the full scan of the genetic neighbourhood, and include sites that match the future predicted climatic conditions. So here we have an example for Melaleuca quinquenervia. If we sample, say for example, from 5 individuals across 5 sites across the full range of the genetic diversity, we're likely to capture high levels of genetic diversity. Now I'm going to add a demonstration on how to use the Restore and Renew web tool at the end of this presentation, but it's super simple. Now I just want to briefly touch on 2 additional ways that you can further maximise the success of your restoration. So one of the ways you can maximise the success of your plantings is to not pull provenances and to maintain maternal lines. So what does this mean? So pooling provenances is when you bring together seed from different sites and put them all in one bag or sow them all out in one tray. And maintaining maternal lines is where you keep the seed from different mother plants separate. In other words, you keep the seed from different plants in separate bags. Now, all the seed from one individual belong to the same maternal line, and so they're all siblings and half siblings. So they share a lot of, so they're very similar. They have a similar genetic makeup. So being able to identify different provenances or material from different sites and from different maternal lines allows you, firstly, and this paper here publication talks about, I think close to a dozen reasons why maintaining maternal lines is advantageous. I'm just going to talk about 2 of them today. But it allows you to mix provenances and maternal lines at planting to maximise outcrossing and minimise inbreeding. For example, here we have 5 plants, seed collected from them, kept separate, maintained in the nursery, and then when these individuals that were propagated were planted out, they're planted out in a way that actually maximises outcrossing and minimises the chances of siblings or closely related individuals being close together. A really simple, effective example of this is Muswellbrook Council currently planting river red gum, source seed from 3 different provenances in order to maximise genetic diversity. And plants are being planted in trios with 1 plant from each of the provenance in each of the trio. Super simple, but a very effective way to maximise outcrossing. Now, this is a more complicated example of doing that. So maintaining maternal lines. This is from a study on the Cumberland Plain, Western Sydney. This is a design for a seed production area for Acacia decurrens, designed to maximise outcrossing in the seed production area. It could equally be a very well-designed restoration site. So in order to maximise genetic diversity, we recommend collecting seed from 5 geographic locations, from 5 different sites across 3 geographic areas: Cumberland Plain, Hunter Valley and Blue Mountains. Some of those sites need to be, we advise some of those sites that are in future climate conditions. And by collecting those individuals, you can capture 90% of total genetic diversity. Keep the seeds separate, both in the seed packets and in the nursery. And then when you plant them out, you can actually make sure or randomise it or design it so that outcrossing is maximised. So whereas if we pooled the seed from different individuals or from different sites, we wouldn't be able to do this. And often in the nursery, the seed from 1 seed packet gets planted out into a tray, then goes on to tube stock, which gets lifted up and planted out at a restoration site. So you are more than likely often to have close siblings just planted together. Whereas if you keep them separate, you can grab seed from the different provenances or different maternal lines and mix them at the restoration site. So going back to the slide on why not to pool provenances and why we should maintain maternal lines, another advantage is that we can isolate hybrid, inbred or planted maternal lines. To give you an example of this, planting of river red gum along Wallenby Brook in the Hunter Valley. Maternal lines were maintained, seed was collected from a dozen mother trees, we genotyped the seedlings, and we discovered a planted tree that looked natural. The stand was naturally recruiting, but what we discovered, and it's, I guess here in the blue is the Wallenby Brook populations. At the top of the circle we have a individual which occurred on the Wallenby Brook, looked natural, but when we genotyped it, we discovered that it was more closely related to individuals on the Lachlan out west. Now, upon further research, we discovered that the property was owned by someone who loved planting native trees. So what it appears like now is that So some river red gums were planted, most likely all very closely related, seed from one individual perhaps, and the seed was so inbred that they have not survived, and all the offspring, the seed that we genotyped from that mother tree, was actually hybrid, and so it was a cross with cabbage gum or Eucalyptus amplifolia. So it demonstrates that if you have a restoration site with very low genetic diversity, you can actually have a individual that produces hybrid seed rather than sorry, rather than pure. And also, importantly, we were able to pull those hybrid seedlings out from the collection and not plant them. Whereas if all the seed had gone into a bag or all into trays at the nursery, pulling out those hybrid individuals would have been like pulling out a needle in the haystack. Now, we also discovered that one of the mother trees, or also another isolated one, looked natural, but it was also planted, and we coincidentally genotyped the mother tree further away at Musselbrook. And what we discovered, the seed from that tree was so inbred, were super inbred. They were all siblings and close relatives. And that seed performed more poorly in the nursery, and germination rates were lower. The group was already, I guess, disappointed that they had to destroy the hybrid plants. So they ended up planting those inbred individuals, but in a way that outcrossing was maximised. They weren't all planted together. And in the future, that packet of seed that we know is inbred won't be used. Now, here's a link there to QR code to a landline story of this project for more information. Now, another way you can further maximise the success of your restoration plantings is to not restrict yourself to sourcing seed only from the target plant community type. Now, in order to get your funding, your plantings need to be a mix of native trees and shrubs, using species that represent the local plant community type. But that doesn't mean you have to source seed only from that particular plant community type. Species occur in more than one community type. Here's a link to a recent research project that was published in Nature Communications. It looked at the relationship between plant community type and genetic relationships for 50 widespread species. And they discovered that while plant community type classification is a convenient way to partition floristic diversity, it actually doesn't reflect the evolutionary history or adaptive potential of single species. So plants don't pay attention to plant community type boundaries. So a key implication for nature repair is that when restoring a particular plant community type, constraining seed collection to only that particular vegetation type for a species is unnecessary and can actually reduce the long-term viability and resilience of the restored vegetation. So, in summary, to maximise the success of your plantings, you want to firstly remember that you're restoring self-sustaining populations and you need to sensibly maximise genetic diversity. You can do this using the Restore and Renew web tool to design your seed sourcing strategy. And you want to grow plants from seed collected from multiple plants across multiple sites across the entire genetic collection area, including sites that match predicted future climatic conditions. Ideally, you'll also keep seed from different sites and ideally different plants separate during propagation so that you can mix them together at planting to maximise outcrossing. And you also don't need to restrict yourself to sourcing seed only from the same plant community type for a particular species. So thank you. Stay in touch. Here's a link to our RISA homepage. If you scroll to the bottom of that page, you can subscribe to our quarterly newsletter, Practitioner Updates from RISA, where we highlight our recent research and its relevance to restoration and conservation. We also publish a publication booklet each year which summarises our publications. It's a very productive team, often 20 or so publications from the year, and abstracts for all the publications each year are in that booklet. You can download that from our publications page. On our publications page, you can also read recent stories from the research team. So thank you. I'll go on now to do the the demonstration of using the Restore and Renew web tool.